Hi everyone, this is Ashley Creighton again. Today I'm going to be talking about Alphafold for my second local expert presentation. So to start off, what is Alphafold? From Alphafold's website, it is an AI system developed by DeepMind that predicts a protein's 3D structure based on its amino acid sequence, and it regularly achieves accuracy competitive with experimental structures. It can be used to help speed up the process of protein discovery, which in turn saves time and money. It can potentially allow scientists to develop disease treatments quicker than what we currently are able to do. And it can be used before and possibly eventually in place of the use of animals to verify protein structures. And then it does link and complement information from other online databases like PDB, which is Protein Data Bank, and Uniprot, which is another protein database online. Okay, so I just want to quickly show you the Alphafold website. So this is the main page. We can go in here and type in our protein. I'll choose MX1. Over here we can search by organism, so we'll just choose Homo sapiens. And then we can also further narrow it down by review status. So if it's reviewed, it's been experimentally confirmed versus the unreviewed. So we're going to choose this first one here. So this is what the protein page will look like. So it gives you general information, source organism, gene, protein. It links directly to the Uniprot and PD websites here, which is super handy if you need information from those websites. You don't have to search specifically. It'll take you directly to their pages for this protein. It has the biological function of the protein. And then if we scroll down, we can see the 3D structure here, which is super cool. The colors do have meaning. So the dark blue, there's a very high confidence in that structure. And then as we get lighter or change to yellow or this orangish color, it gets less confident in the structure. So this is a really good example that kind of has each of the different colors. There are other ones that have very high confidence, so we can look at hemoglobin. So as we can see for hemoglobin, it is actually almost completely confident in the structure except for this last spot right here. If we go back to our MX1 protein, we can go over here and there's a lot of different things that you can do to manipulate this image here. So if we go here, we can actually add different views, add representations. So we can highlight the backbone. And it's hard to see zoomed out, but if we kind of zoom in here, this green line here is that backbone of this structure. We can go in here and look at the ball and stick, which is kind of hard to see everything, but it can be helpful, especially if you're looking at specific amino acids. We can also hide those or completely get rid of those views too. Another one that can be super helpful is looking at the surface of the structure too, because it's one thing to look at like the backbone and the ball and stick views, but the outer structure really can be really helpful. Also, if you want to look at a very specific amino acid, you can go here and look at the chain and then choose that amino acid and it'll zoom into it for you and show you that ball and stick view. Or if you just want to look at this amino acid right here, you can click on that and it will show you the same view. So I can't talk about Alphafold without talking also about CASP. That stands for the Critical Assessment of Protein Structure Prediction. So this is basically a biennial experimental competition in the scientific community to look at different protein structures and prediction methods and how they compare to the actual experimental protein structures and each other's methods. The next one is going to be CASP 16, and it's coming up in 2024, so sometime next year. So the way it works, predictors are provided with an amino acid sequence of the target protein, and their job is to build a 3D model with whatever methods they choose. And then there are assessors that compare those models with the experimental structures. So they've gone through and done all the different techniques experimentally to confirm those structures. It is double-blind, so the participants don't know what the actual structure of the protein looks like before they start, and the assessors don't know who submitted the model predictions at the end that they're assessing. So that kind of allows for better results because there isn't any prior knowledge to those structures before they start. The results are all assessed by committees based on model accuracy and other different criteria that they've developed over the years. In CAS 14, back in, that would be 2022, so last year, Alphafold was actually the top ranked prediction method in terms of its accuracy and speed of being able to develop those models. Currently, Alphafold is mainly used for single domain protein structures. There are a couple other AI programs out there right now that are able to predict larger protein structures and proteins that might have multiple domains that interact with each other. One example of this is deep assembly, which I'll actually be going in and showing you an example of later. Alphafold isn't perfect, but it can help speed up our scientific processes by giving researchers a starting point for their protein structures before they actually start doing the experimental confirmations. The predicted structures do still need to be confirmed experimentally because right now they're not 100% accurate with the current models and methods. So confirmation can be done using crystallization or X-ray diffraction techniques, both of which can take a really long time, and because of that time alone can be more expensive, but also you have to think about the equipment needed for those techniques also. Alphafold does have some other limitations that should be considered when you're trying to use it for predictions. The length of the amino acid chain shouldn't be too long or too short. On their website, they do have some general guidelines for the ranges. And it the amino acid sequence shouldn't contain any non-standard amino acids. And it currently can't do anything with viral proteins. All of these are areas that can, and they're actually currently being worked on for improvement so that eventually those constraints won't be a problem anymore. So this image from my first manuscript is actually showing a comparison of 3 different protein structures, comparing the Alphafold prediction to the experimental structures. So in green, we can see the experimental structures, and then it's overlapped with the blue Alphafold predictions. So we can see overall the predictions pretty closely match the experimental structures, but there are a couple spots in each of these proteins that the Alphafold and experimental structures don't actually match. So that's where it's important to still do that experimental confirmation of the predictions. So in my second manuscript, this study was actually using a similar program called deep assembly to predict structures of proteins with multiple domain subunits, so larger proteins. In this image specifically, we can see in the gray, they have the experimental structures of transmembrane proteins from PDB, and it is being compared to the predicted structures from deep assembly. The different colors in each of the structures show the different domains or the chains of each of the proteins. So in this last study, they were looking at protein ligand binding sites to eventually be used in drug discovery experiments. In the blue, there's the Alphafold predictions, and then the gray is the experimental structures that they got from PDB. As we can see, just like the other ones, there's a lot of matching, but there are a few sites that they do differ from each other. Thank you for listening to my talk about Alphafold, and here's a list of resources that I used in case you want to learn more.